Yeah, so it's not stretches.
What?
Where's the doggy?
The dog? In this crate where he always is. That's where he stays when y'all are here. Some things don't change.
I think he deserves a second chance.
Not willing to take that.
What's that? Views.
All right. Y'all got it. Are we done? We have one more. One more? Almost as you. Yeah. Okay. I don't know about her at all. Becky or Fallonja. No, like he's not me. Okay. I follow Marvel stuff. That's why you're being Scarlett Johansson related emails. That's where I feel. All right.
Well, if you invert it, then it's worse. It's greater than.
Next week, we're going to very much abbreviate lab so that we can spend a lot of time on review.
One piece. Yeah, that's why I asked if you guys are involved. No, I'm not. It looks like it's. So it's supposed to be in the other. Oh, you see why it's teaching. Yeah, that'll be also better. No. Yeah.
Of course.
What? I'm using the abbreviation. What do you mean?
So have you seen, I was talking to Jackie about this morning, have you seen the fan series about Nettie coming back as cat?
Wrong abbreviation.
I don't know anything about Dungeons and Dragons, but apparently there's a character named Cass who's like some kind of vampire and he's like an acolyte.
I have seen
that. But there's this whole stuff where people are like, Eddie was killed by vampires. Eddie looks like Cass, like all this stuff. And I'm like, I don't know he's dead, which makes me very spoilers. No, we don't know that. We know that.
That was like 3 years ago, Isabella.
Oh, that was a year ago. Where you been?
Just started watching.
I gotta catch up. All right.
No, it is. I doubt it because his actor didn't return. He said that the actor never came back. So
Yeah.
no actor, no filming.
Although I did see a video of him playing.
Was it AI though?
Yeah, no, I saw a video of him, the actor.
Oh, okay.
Metallica playing guitar with him.
Oh,
That was
that's
awesome.
awesome.
All right, well, juniors, you guys are good. You can head on out. If you have finished and you know that it's right and you're happy with it, you can turn it in. If not, you can take it home and keep working on it, all right? Okie dokie, gang.
I should just give this to you.
Oh, yes, you are.
Let me give this to you.
Thank you. All right, I'll let everybody get situated here. Juniors, study for your midterm. Start your midterm studying if you haven't already. Don't cram. Better to study ahead than to cram. We can do a little last minute cram.
Don't cram in college either, because guess what I'm doing next? Cramming, because that's my history final when I get home.
Not fun, is it? Study as you go, and if you need a last minute cram, you can last minute cram, but
That's why I brought
try to avoid it.
That's why I brought it.
There you go. All right, so we are talking today about population evolution. So we talked about how genetic mutations can cause a change in an allele, and then that could get passed on, and you can get some degree of evolution. But the biggest point of evolution is really within a population, not within individuals. So Barrett, are you doing notes or something? If you're not, pay attention, okay? Because we want to be sure. Here's the thing. You guys are going to be studying for your Unit 6 test and your midterm simultaneously. Your unit 6 test is due Monday, and then you walk in and do your midterm. So you have to know what's going on. You don't want to last minute cram for any of it. You got plenty of time to prepare and study. All right, so let's think about it a bit. Darwin developed his theory of evolution without knowing how heritable traits are passed from one generation to the next, or where any heritable variation came from. So this bit of science wasn't known yet, but he still formed his theories. So then what would happen when genetics answered questions about how heredity works? Well, for the most part, proved that he was on the right track, as we'll see. So when genetics joined evolutionary theory, we get, how is evolution defined in genetic terms? Does anybody know that one for me? Caitlin.
Hold on.
Yeah? Pause. I love how your generation uses pause just like wait, because y'all have been on phones and computers for so long that pause is like normal for you to say. Yeah. She's looking for it.
Nope, I don't have it.
That's fine. All right, so it's a change in the relative frequency of alleles in the gene pool. Okay, so we'll talk about allele frequency here, but when allele frequency changes, that's when you see evolution in like in progress, working. So how do mutations affect allele frequencies? So at the start of the human race, brown was the only eye color that people could have. Allele frequency for that allele was 1 or 100%. And then a mutation in the main gene for eye color created a new phenotype, which was blue eyes, and slowly the allele frequency shifted. Since then, we've gotten lots more variations in eye color, though brown still is the most common. So within the gene pool, we have more alleles for brown eyes than blue eyes, but we've had a shift because it used to be 100% brown. So researchers discovered that heritable traits are controlled by what? What controls our heritable traits? Genes, yeah, he's like, is this the right answer? Yeah, it's genetics, genes. And changes in genes and chromosomes generate what? What change, if you change a gene or a chromosome, what happens? You get variation in your population. So for example, all these children receive genes from the same parents, but they all look different, right? Everybody, unless you're an identical twin, you're going to look different, even from your siblings, even though you're coming from the same very small gene pool of two parents. So genotype and phenotype in evolution. So what is the oh shoot. Okay, I just got a plug. Plug in the plug. I don't know what does that sometimes, it just gets funky. There it goes. So what do you call the combination of alleles that an organism carries? That's its what? The combination of alleles is its, Jordan? Not gene pool, but you're close. You're real close. Isabella? Not genome. You're close too. The combination is your genotype, right? Okay. And then your genotype and the environmental conditions produce your, yes, Lucas, phenotype. Very good. All right. So phenotype includes all physical, physiological, and behavioral characteristics of an organism. Natural selection acts on what? Doesn't act on genotype. It acts on, yeah, it acts on your phenotype. Okay. Some individuals have phenotypes that are better suited to the environment than others. These individuals produce more offspring, pass on more copies of their genes to the next generation, right? That's how it works. We get that. So what is a group of individuals of the same species? Hang on, is this one of the boys texting me that they can't? If you see Yusuf or Eunice dinging up here, let me know because they might show up. Anyway, what is a group of individuals of the same species that can mate and produce offspring? What do we call them? Think back to our ecology lessons at the beginning. The group of a species is what? Yes, population. Good. That's your population. Okay. And then what do we call it when you have all of the genes from all of the alleles in a given population? You put all the genes, all the alleles from everybody in the population. What do you call that, Caitlin? Yes, gene pool. Good. All right. So that's your gene pool. Your gene pool is all the alleles, all the genes, everybody in the population. And then researchers study gene pools by examining how, does anybody know this one? So this is your relative frequency of your allele. So your relative frequency of alleles is the number of times a particular allele occurs in a gene pool compared with other alleles. All right, we have how many frogs? 15 frogs. How many red frogs?
One.
So what is the frequency of red frog phenotype? 1 in 15 or 1/15, right? How about purple frogs? 2. And how about green frogs? 12/15 or 12 out of 15. And you could make that into a percentage how?
Decimal.
Yeah, you're right. How would you make 12/15 into a percentage?
You would divide 100 by 15 and then
You divide
percentage and you multiply.
You divide 12 by 15, get a decimal, move your decimal back 2 places, right? Multiply it by 100 to get your percent, right? 2 over 10 is what percent?
1/5.
What percent?
Oh,
20%.
yeah.
There you go. All right. So, what is any change in the relative frequency of alleles in a gene pool of a population over time? What do we call it when there's a change in the relative frequency of alleles in the gene pool? That's our evolution, okay? So if you get, it's not about one individual having a mutation and changing. It's about the relative frequency of the allele in the gene pool that starts an evolutionary process. So natural selection operates on individuals, but resulting changes in allele frequencies show up in populations. So this is the big takeaway. Populations evolve, not individuals. Populations rather than individuals evolve. So say these are frogs again, and we started with the population where 80% of the alleles were for green frogs and 20% were for brown frogs. And over the course of a year, you had 60% for green frogs and 40% for brown frogs. There has been some degree of evolution within the population. What might cause that?
Mutation.
Well, you've already got both alleles in there. What do you think, Isabella?
Natural selection.
Natural selection acting how? What do you think?
Predator.
Say it again?
Predator. Mostly.
Predators. Okay. What were you going to say?
Change in habitat.
Change in habitat? Absolutely. Yes. Something else? No, same thing? All right. So that's what we're looking at here. All right. Now, what are the sources of genetic variation? We have 3 big ones. Do you know what they are? Caitlin? Nope. All right. So what we're looking for is mutation, genetic recombination during sexual reproduction, and lateral gene transfer. Okay, so these are your big sources. We're going to do 3 more that affect more of a population evolution. This is more like the individual, what causes the individual to change. So genetic mutation, genetic recombination during sexual reproduction, lateral gene transfer. Don't worry about those as much. What we really are going to focus on when we get to the population part are the things that affect population evolution. So mutations that produce changes in a phenotype may or may not affect fitness. We've talked about this. Some are lethal, some lower fitness, others are beneficial, right? So not all mutations are bad. Mutations matter in evolution only if they can be passed from generation to generation. So for that to happen, your mutation has to occur in your germline cells, or what we call the cells that produce your gametes, the cells that produce sperm and eggs. If the mutation occurs there, then it can be passed. If you get mutations in, some other part of your body, you could, maybe have a growth or something that would happen, but that's not necessarily going to get passed on, right? As it's showing here. It has to be in your germline cell in order for it to make any effect on offspring. All right, so this is great where it turns genes into genes, like the kind you wear, and it shows how the shuffling works, and it shows how you can have evolution of a population, and they're using redheads as the example. So I don't know why, but it's actually kind of cute.
Bill dropped.
Yeah, energy bill. Okay.
Doing this.
Ads.
$49.
Here you go.
Yep.
Yep.
The 5 fingers of evolution. A thorough understanding of biology requires a thorough understanding of the process of evolution. Most people are familiar with the process of natural selection. However, this is just one of 5 processes that can result in evolution. Before we discuss all 5 of these processes, we should define evolution. Evolution is simply change in the gene pool over time. But what is a gene pool? And for that matter, what is a gene? Before spending any more time on genetics, let us begin with a story. Imagine that a boat capsizes and 10 survivors swim to shore on a deserted island. They are never rescued and they form a new population that exists for thousands of years. Strangely enough, 5 of the survivors have red hair. Red hair is created when a person inherits 2 copies of the red gene from their parents. If you only have 1 copy of the gene, you won't have red hair. To make this easier, we will assume that the 5 non-redheads are not carriers of the gene. The initial frequency of the red hair gene is therefore 50%, or 10 of 20 total genes. These genes are the gene pool. The 20 different genes are like cards in a deck that keep getting reshuffled with each new generation. Sex is simply a reshuffling of the genetic deck. The cards are reshuffled and passed to the next generation. The deck remains the same, 50% red. The genes are reshuffled and passed to the next generation. The gene pool remains the same, 50% red. Even though the population may grow in size over time, the frequency should stay at about 50%. If this frequency ever varies, then evolution has occurred. Evolution is simply change in the gene pool over time. Think about it in terms of the cards. If the frequency of the cards in the deck ever changes, evolution has occurred. There are 5 processes that can cause the frequency to change. To remember these processes, we will use the fingers on your hand, starting from the little finger and moving to the thumb.
This is a great one to memorize.
Remind you that the population can shrink. If the population shrinks, then chance can take over. For example, if only 4 individuals survive an epidemic, then their genes will represent the new gene pool. The next finger is the ring finger. This finger should remind you of mating because a ring represents a couple. If individuals choose a mate based on their appearance or location, the frequency may change. If redheaded individuals only mate with redheaded individuals, they could eventually form a new population. If no one ever mates with redheaded individuals, these genes could decrease. The next finger is the middle finger. The M in the middle finger should remind you of the M in the word mutation. If a new gene is added through mutation, it can affect the frequency. Imagine a gene mutation creates a new color of hair. This would obviously change the frequency in the gene pool. The pointer finger should remind you of movement. If new individuals flow into an area or immigrate, the frequency will change. If individuals flow out of an area or emigrate, then the frequency will change. In science, we refer to this movement as gene flow. All 4 of the processes represented by our fingers can cause evolution. Small population size, non-random mating, mutations, and gene flow. However, none of them lead to adaptation. Natural selection is the only process that creates organisms better adapted to their local environment. I use the thumb to remember this process. Nature votes thumbs up for adaptations that will do well in their environment and thumbs down to adaptations that will do poorly. The genes for individuals that are not adapted for their environment will gradually be replaced by those that are better adapted. Red hair is an example of one of these adaptations. Red hair is an advantage in the northern climates because the fair skin allowed ancestors to absorb more light and synthesize more vitamin D. Thumbs up. However, this was a disadvantage in the more southern climates where increased UV radiation led to cancer and decreased fertility. Thumbs down. Even the thumb itself is an adaptation formed through the process of natural selection. The evolution that we have described is referred to as microevolution because it refers to a small change. However, this form of evolution may eventually lead to macroevolution or speciation. Every organism on the planet shares ancestry with a single common ancestor. All living organisms on the planet are connected back in time through the process of evolution. Take a look at your own hand. It's an engineering masterpiece that was created by the 5 processes I just described over millions and millions of years. Can you recall the 5 main causes of evolution from memory?
Okay, can you? What's the little one mean?
Small.
Yep, populations can shrink. What does the ring finger mean? Non-random mating. And the middle finger? Mutations. What's the pointer? Gene flow. And what's the thumb? Adaptations, yes or no? See, it's great. Very good. And you can actually see, if you want to see that small, the population size one in effect, go on niche and look up small towns and compare them to large towns, because niche will do demographics. They'll do age and sex demographics and religious demographics and ethnic demographics for every town. And in large towns, you'll find the demographic is similar to the national average, but in small towns, you might have something like, if a town has 1,000 people in it, you might find something like 60% female, which is way beyond what like the 51 that you would expect to see. So you can actually see that in smaller populations of humans. All right, so insect populations contain a few individuals that are resistant to a particular pesticide. And then those pass on their resistance to their offspring, and soon the pesticide-resistant offspring dominate the population. And the relationship between natural selection and genetics explains how pesticide resistance develops. This is the same thing that happens with antibiotics. MRSA, which is a antibiotic-resistant strep type of disorder, strep bacteria. Yeah, we have all kinds of bacteria that have become resistant to antibiotics because we use them a lot. This is also why you shouldn't take antibiotics unless you absolutely need them, because not just because it's not going to help you, but because population-wise, you're exposing more bacteria to that antibiotic, and the ones that are resistant to it are going to thrive, and there become more of them in the population. All right, so how does it work? How does natural selection affect single trait and polygenic traits? So natural selection on single gene traits leads to changes in allele frequencies and thus changes to phenotype frequencies. Natural selection on polygenic traits can affect, they're waiting for me to let them in, but I don't see them here. Hold on. Okay. Hold on, I want to be let in. Shh, I love it. But they're not here. Ugh. Shoot, I don't see them. Nobody sees them, right? You don't see them anywhere that I'm not seeing them.
That's fine.
I don't see you. Oh, well. Okay, so anyway, as we were saying, natural selection of phenotypes, feel terrible just leaving them out, but I can't find them. Natural selection on polygenic traits. Remember what polygenic traits are? What are polygenic traits? When you have multiple genes that affect a single trait, like height, right? Can affect distributions of phenotypes in 3 ways. Caitlin, you got it?
Directional, stabilizing, distributive.
Yeah, okay. So directional, stabilizing, or distributive selection. So how does that work? Evolutionary fitness is the success in passing genes to the next generation. Evolutionary adaptation is any genetically controlled trait that increases an individual's ability to pass along its alleles. So natural selection for a single gene trait can lead to changes in the allele frequencies and then to evolution. So for example, a mutation in one gene that determines body color in lizards can affect their lifespan. So the normal color for lizards is brown. A mutation may produce red and black forms. Whichever one of those produces a longer lifespan is likely to continue and be part of the new gene pool. So directional selection. So Caitlin mentioned these different types. Directional selection occurs when individuals at one end of the curve have what? What fitness? Better fitness? Less fitness?
Better.
Yeah, higher fitness. Very good. Higher fitness or better fitness than individuals in the middle or at the other end. The range of phenotypes shifts because some individuals are more successful at surviving and reproducing than others. So, for example, if only large seeds were available, birds with larger beaks. Hey, where did we do this? Birds with larger beaks would have an easier time feeding and would be more successful in surviving and passing on genes. Click the live stream on that. I know. Yeah, I know. I know. Hold on. Hang on, let me just. It's probably going to be better to just watch it later, period. I'm sorry, period. I'll troubleshoot later. Okay, they're trying, but I can't. She's texting me. Okay, all right. There. All right, so, for example, if only large seeds were available, birds with larger beaks would have an easier time feeding and would be more successful in surviving and passing on genes, right? We saw this. So stabilizing selection occur. I cannot read today. When glasses are terrible. When individuals near which end of the curve? Caitlin. Near the center, very good, of the curve have higher fitness than individuals at either end. And this situation keeps the center of the curve at its current position, but narrows the overall graph. So for example, very small and very large babies are less likely to survive than average sized individuals. And the fitness of these smaller or larger babies is therefore lower than that of more average sized individuals. Why might larger and smaller babies have a harder time surviving? What do we think?
Larger
Mm-hmm.
babies require more food.
There you go.
Smaller ones are more likely to die by cold.
There you go. That's a perfect answer. Yeah. So large babies may require more food than is available to them, and smaller ones may, have issues with body temperature maintenance or other things. Uh-huh.
There was a baby that was addicted to smoke,
Mm-hmm.
and then they weaned the baby off of the smoke addiction, but then the baby got addicted to food.
What kind of smoke? Like smoking?
Smoking.
Oh, like a toddler?
Yeah.
Oh, like a toddler was smoking, like in some country where that's not so weird. Yeah, okay. Yeah. I was gonna say.
Don't you mean like campfire smoke?
Yeah.
I think I'm moving.
Well, that can be a thing too. If you've developed a certain tendency towards addictive behaviors, you can be more inclined to do, and some of that is genetic and some of that is habitual, but you can be more inclined to become addicted to all sorts of things, right? All right, so disruptive selection occurs when individuals at which part of the curve? There you go. At the upper and lower ends of the curve have higher fitness than individuals near the middle. So disruptive selection acts against individuals of an intermediate type and can create two distinct phenotypes. So for example, in an area where medium-sized seeds are less common, birds with unusually small or large beaks would have higher fitness. Therefore, the population might split into two groups, one with smaller beaks and one with larger beaks. But it says population, so we're still the same species, right? There's no speciation here. So what's genetic drift? So in small populations, individuals that carry a particular allele may leave more descendants than other individuals just by chance. Over time, a series of chance occurrences can cause an allele to become more or less common in a population. So this is really a thing for smaller populations, because in larger populations, it all just kind of balances itself out. But in smaller populations, you can have that chance situation where suddenly you have more. Again, like I said, the small town demographic situation. And a genetic bottleneck is your change in allele frequency following a dramatic what in the size? What do you think, Kenna? There you go. A dramatic reduction in the size of population. So, for example, a disaster may kill individuals in a population, and the surviving population's gene pool may contain different gene frequencies from the original gene pool. That makes sense, right? Like if you think back to the little genes on the island, it even showed you like, oh, there's some terrible event that occurs, and then you only have these left. And these are the only ones whose genes are now in the gene pool because the others died in whatever terrible cataclysmic event. So then we have something called the founder effect. And this occurs when allele frequencies change as a result of the migration of the small subgroup of a population. So two groups from a large diverse population could produce new populations that differ from the original group. So if they started out together, but a small group of them migrated away, you can get populations that become more different. And you see this sometimes with like coloring of animals that live in certain areas. You can sometimes see a difference, like those that live closer to this geographic feature have slightly different coloring than those that live closer to this geographic feature. So it's something you can see. Evolution versus genetic equilibrium. So what conditions are required to maintain genetic equilibrium? So we have this thing called the Hardy-Weinberg principle. And there are 5 conditions required to maintain genetic equilibrium. Now, this is good for prediction. This is one of those prediction tools that we use, kind of like, for those of you who did chemistry, do you remember ideal gas law? Like, is an ideal gas a thing? What is an ideal gas? Does anybody remember? It's like, do you remember, Asher?
It's a very unreactive.
Yeah, it's like in a perfect condition, this is what we would have. But realistically speaking, nah. So here's why this is good for prediction. So the population must be very large. All right, you can do that most of the time, but not all of the time. 2, there can be no mutations. Is that going to happen? No, because we're always going to be mutating things. There must be random mating. Is that going to happen? No, because females are typically selective and choosy, and your non-fancy males or your non-strong males or whatever are not going to get any mates. There can be no movement into or out of the population. Okay, maybe that could be something you could control for. And then there's no natural selection. Is that going to happen? No. All right, so a population is in genetic equilibrium if allele frequencies are just staying the same. They don't change. A population does not evolve. So this is the idea behind this principle. So the Hardy-Weinberg principle describes the conditions under which evolution does not occur. So allele frequencies in a population remain constant unless one or more factors cause those frequencies to change. So here we go. Great explanation using frogs again. Frogs are the theme of the day, apparently. Frogs and birds. I don't know why, but
my energy bill dropped from 100.
This gets mathy, but it's okay.
Friends laughed at me when I spent $49.
You know what? My friends are AI.
Yeah, there's that.
Do you like math? Sometimes when people are starting out in biology, for some reason they assume there is no math in biology, but then they soon find out they were wrong. My math teachers were telling the truth when they said math isn't everything. Chi-squares, osmotic pressure calculations, standard curves with gel electrophoresis, Punnett square ratios, these all have math. But one of my favorite topics that involves biology and math, and is also absolutely fascinating, is the Hardy-Weinberg equilibrium. It got its name from a mathematician and a physician, and it states that a population's allele and genotype frequencies are constant, unless there's some type of evolutionary force acting upon them. A reminder from our ecology videos, a population in this case is a group of organisms that are all the same species and can breed with each other, and have fertile offspring. However, there can be variety among a population as they're not clones of each other. But based on Hardy-Weinberg equilibrium, the population's allele and genotype frequencies would remain constant if no evolutionary force acts upon them. If we have these frogs here, assume they are all the same species in this example, but there are some slight differences. In our simplified example, some of the frogs are lighter green and some are darker green. We're going to use a simple genotype in this example. All frogs in this population will be either big G, little g, or little g. There's also an allele frequency in our population. We're going to say here that 60%, a frequency of 0.6 of the alleles are big G. 40%, or a frequency of 0.4 of the alleles are little g. Notice the percentages add up to 100%, and the frequencies add up to 1. Now, to be in Hardy-Weinberg equilibrium, we have to have 5 assumptions. Number 1, no selection. No natural selection is acting upon these frogs.
That means neither dark green nor light green will have any impact on reproductive fitness. Number 2, no mutation. Baby frogs inherit genes from their parents and there are never mutations. Number 3, no migration. Frogs can't come in, frogs can't go out. Number 4, large population. There are a lot of frogs. It turns out small populations are more vulnerable to genetic drift, by the way. See our genetic drift video. Number 5, random mating. The frogs mate without any specific choice. All of these 5 assumptions must be kept in order for Hardy-Weinberg equilibrium to happen. So in real life, does this generally happen? No. For example, in real life, maybe in a certain environment, predators can more easily see the light green frogs, so perhaps they are eaten more and have less reproductive fitness. So if Hardy-Weinberg equilibrium is unrealistic in nature, why does the Hardy-Weinberg equilibrium matter? That's where the math comes in. The Hardy-Weinberg equilibrium gives you this baseline to compare how an evolving population could compare to one that remains constant without evolutionary forces acting upon it. And now let's explore the math. So there are 2 equations in Hardy-Weinberg equilibrium that we'll focus on. We're going to start with the first one. P plus Q equals 1. In this equation, P is the dominant allele frequency in the population, and Q is the recessive allele frequency in the population. By the way, Hardy-Weinberg equilibrium doesn't mean that P has to equal Q, and the dominant allele frequency in any population doesn't have to be larger than the recessive allele frequency in a population. That's a misconception because dominant alleles aren't always the more common allele. The equation does say that the dominant allele frequency and the recessive allele frequency have to equal 1. So in my example here, if I say 60% of the alleles are big G, 40% of the alleles are little g, 0.6 is P, and 0.4 is Q. So this equation, P plus Q equals 1, is for those allele frequencies. But what if I wanted to know the genotype frequencies? Meaning, I wanted to know the frequency of frogs that are homozygous dominant, which is big G, heterozygous, which is big G, little g, or homozygous recessive, which is little g. Then I can use this other Hardy-Weinberg equilibrium equation, P squared plus 2PQ plus Q squared is equal to 1. So P squared is the homozygous dominant frequency, the frequency of big G in this case. 2PQ is the heterozygous frequency, so big G frequency in this case. Q squared is the homozygous recessive frequency, so little g frequency in this case. Let's plug those previous P and Q values in. P squared would then be 0.36. 2PQ would be 0.48. Q squared equals 0.16. Cool, huh? The only thing is that when calculating Hardy-Weinberg equilibrium problems, you don't always have the P and Q values. So we're going to do an example. New population of frogs, so forget the previous frequencies. This is a new frog population in a new frog land, but we'll still use the same allele letters. So here's the information you get for this new population. There are 500 frogs, and of those, 375 frogs are dark green. The rest are light green. With that information, please solve all genotype frequencies and all allele frequencies if in Hardy-Weinberg equilibrium. Step 1, determine first whether you're going to work with the first equation or the second equation. Since I'm working with individuals here that have genotypes, I'm going to work with the second equation first. Step 2, figure out what value you can determine. So 375 frogs are dark green out of 500 total. That would mean 125 frogs are light green since there are 500 frogs total. But I can't use full numbers like that, as both of these equations ultimately are equal to 1. I need frequencies for these equations. Now I could say 375 dark green frogs out of 500 total frogs is 0.75, but all I know is that would be the frequency of dark green frogs. The problem is dark green frogs could have genotype big G, or they could have big G. I can't assume they're one genotype or the other, so I shouldn't use that value. The recessive genotype, resulting in a light green trait, is safe to use though, because I know that light green frogs are the genotype little g. They can't be anything else. So 125 frogs out of 500 frogs equals 0.25. That means from the equation, the value q squared equals 0.25. Step 3, take a value you solved from the previous step and calculate from there. So if I know that q squared is 0.25, then I could go ahead and solve for q. If q squared is 0.25, I can determine q if I take the square root of 0.25. Therefore, q is equal to 0.5. That's the allele frequency for the recessive allele little g. If I know the q value, I can find out the p value using the first equation. Since p plus q is equal to 1, now I know that p is equal to 0.5. That's the allele frequency for the dominant allele, big G. Now I have p and I have q, and I can take care of everything else. I can use the second equation to determine the homozygous dominant frequency, the heterozygous frequency, and I already knew the homozygous recessive frequency from the beginning. Plugging those in, p squared equals 0.25 for the genotype frequency big G, 2pq equals 0.5 for the genotype frequency big G, q squared equals 0.25 for the genotype frequency little g. Phew, that's a lot. Finally, some last tips for solving Hardy-Weinberg equations. Number 1, I used nice numbers, meaning I didn't need a calculator for my examples. It's not always like that. You might need a calculator for the numbers you're given, and you might need a round. Number 2, when you get your final results, always check that the values you calculated do equal 1 for both equations. This can really help you check your work. Number 3, be careful not to assume too much with given info. Remember when I was given a value of 375 frogs out of 500 being dark green? I didn't necessarily know whether they were big G, or big G, little g. Instead, I chose to work with the recessive genotype since I knew light green frogs would be little g. Number 4, practice. There are a lot of practice problems you can find online, and remember, this skill can be very useful for determining the extent of evolutionary forces because you have this base comparison of Hardy-Weinberg equilibrium to compare it to. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So, should you take an intro bio course in college, even one for non-STEM majors, that may be thrown at you. And I know because Cole called me freshman year and said, I have a very vague memory of this Hardy-Weinberg and I have this problem and I'm stuck. So if you get there and you find yourself in that situation, remember that Amoeba Sisters has a really good video, right? That should refresh whatever you don't remember because that will come up again, I promise you. In this class, not too much. Beyond this class, yes. So be aware of it. It's a thing. All right, so now let's talk about large population. So what can cause changes in allele frequencies in small populations? Genetic drift. Genetic drift has less an effect on these large populations because large population size helps maintain genetic equilibrium. And then no mutations, as we said, not really a thing. If mutation occurs, new what can be introduced to the gene pool? What could happen? Caitlin? Yeah, new traits, yeah, new alleles, okay, or can be introduced to the gene pool, and then that's going to change your allele frequencies, obviously, because you have new ones being added. Random mating. All members of populations must have equal opportunity to produce offspring. Individuals must mate with other members of that population at random. That does not happen in nature, for sure. Doesn't happen with humans, doesn't happen with random animals out in nature either. In natural populations, however, mating is not random. So female peacocks, for example, choose, and actually they're peahens, really, right? They choose mates on a basis of their physical characteristics, such as their brightly colored tail feathers, and such non-random mating means that alleles for those traits are under selection pressure. The fancier your tail, the more likely you are to get a date, right? So no movement into or out of the population. Individuals who join a population may introduce sure. Alleles, new alleles to the population. Individuals who leave may remove alleles from the gene pool. Thus, no alleles, for no alleles to flow into or out, there must be no movement of individuals into or out. And that's not going to happen very often. This you could maybe force to happen based on geographic or structural situations you've got going on. But typically, critters are moving. And then no natural selection. All genotypes in a population must have equal probabilities of surviving and reproducing. No phenotype can have selective advantage over another. And we know that doesn't happen. But it's great to use for the purpose of prediction or understanding how many alleles you have in a gene pool. So meiosis and fertilization do not change the relative frequency of alleles in a population. The shuffling of genes during sexual reproduction produces many different gene combinations, but does not alter the relative frequencies of alleles in a population. Think back to the video with the genes and the deck of cards being shuffled, right? You're continuing to shuffle, but you're not changing the frequency of the alleles. So 5 factors for evolution: genetic drift, allele frequencies change due to chance, gene flow, movement of alleles from one population to another, mutation, new alleles form, sexual selection, traits improve mating success, and natural selection. So if you look at it, this is the pinky again, right? Smaller populations are going to be greater impacted, right? Gene flow, this is your pointer finger, things moving around. The mutation was your middle finger, right? Sexual selection was your ring finger, and then natural selection was your thumb. Again, different way of stating basically the same concepts. All right, so now let's talk. What's a species? Anybody know what a species is? How do we define a species? Yeah. Similar organisms. What do they have to be able to do to be a species? Drew?
Able to reproduce with each other.
Reproduce. And what does the offspring have to be able to do? Reproduce, right. So a group of living organisms consisting of similar individuals capable of exchanging genes or interbreeding. Species is principal natural taxonomic unit, ranking below genus and denoted in Latin by binomial nomenclature. So this says Lycorma delicatula. I cannot see. And so this would be your genus and this would be your species. And for it to be a species, the offspring has to be fertile, right? We talked about zonkies. You can get a zonkie by taking a zebra and a donkey and breeding them to one another, but that zonkie cannot reproduce. Same thing with mules. They cannot reproduce. Yeah.
What about a donkey and a dragon? How does that work?
That would be cool, wouldn't it? Drunky. Yeah. Zagging. Or no, donkey, dragon. No, just be dragon. Yeah. So speciation is the formation of a new species. And as new species evolve, populations become reproductively isolated from one another. Members of 2 populations cannot interbreed and produce fertile offspring. Then what do we say has happened? Reproductive isolation, right? And reproductive isolation is necessary for speciation. So when populations become reproductively isolated, they can evolve into 2 separate species. Reproductive isolation can develop in a variety of ways, including behavioral isolation, geographic isolation, and temporal isolation. And we're going to talk about those. Speciation in the Galapagos finches most likely occurred by founding of a new population, geographic isolation, changes in the new population's gene pool, behavioral isolation, and ecological competition. So, behavioral isolation. This is when you have 2 populations that are capable of interbreeding, but they have differences in their courtship rituals or other types of behavior. All right, you've seen the birds, right? The birds that dance. Has everybody seen the videos of the birds that dance to attract the girl birds, the boy birds? Like the bower bird. Have you seen the bower bird? Like he builds the nest and puts all of his little treats that he finds in it. And you've seen the birds that like, you know what I mean? And they dance. If for some reason all of the female birds in a group don't find that at all attractive and they would rather he stomp his feet than shake his head, then they're going to ignore them and they're going to be like, no, sorry, dudes, you are not mating with us. Go shake your head over there. I want the dude who stomps his feet. So that's a behavioral choice that they're making. And that can happen. That absolutely can happen. That happens a lot with humans too, obviously, because you have culture impacting things.
Stomp their feet.
Right? So awesome.
So hot.
Yeah, it is. Then you have geographic isolation. This one's the easiest to understand. You've got barriers, mountains, rivers, oceans. Look at Australia. Look at the variety of living things in Australia. Everything tries to kill you there, right?
Spider.
Yeah, new spider every week. A whole ton of animals that look nothing like the rest of the world because they are geographically isolated. So speciation over there has been impacted by that isolation. They can't, the mammals that are there cannot breed with the mammals in Asia or Europe or Africa, and therefore you get completely funky new animals. So geographic barriers. Now, temporal isolation. What's the word temporal mean? Jordan.
Rules like passage of time.
Yes, it does. Deals with time. Perfect. Okay. So this has to do when reproduction cycles differ. Okay. How many opportunities do humans have to reproduce in a year? Typically speaking, your typical human female has how many opportunities to reproduce in a year?
Isn't it like 30?
Nope, it's not 30. 12, yeah, roughly 12 every year. 12 opportunities to reproduce within a year. That's great. That makes it easy, right? Now, dogs.
Once a month.
Dogs are typically maybe 2. Dogs have an estrous cycle, not a menstrual cycle, so it's probably closer to 2. Some organisms only are fertile once a year. Like, in the fall when it's deer rutting season and they're all out running in the roads and all the bucks are running around like, ah, have you seen them? They get crazy. Like they run around, they can smell all the females. The females are all in their heat cycle and they're like, this is it. This is my time of year. And they are in the road and they get hit. Like there are more dead deer on the road in the fall because they're out because it's rutting season. That's what they do. This is our time of year. So they are out, right? And running around like crazy things, right? But if for some reason there was a shift in cycle, in their reproductive cycle, where it became winter or summer or spring, then you're going to get one population that is breeding this time of year and another that's breeding that time of year. And that's how you get a shift that's a temporal shift that can cause some reproductive isolation. All right, so we have all kinds of things that impact speciation, and I want you to know about these, but we're not going to be crazy about memorizing them all. But you have allopatric, peripatric, and sympatric. And there's a list here to kind of explain them to you. So allopatric is basically when you have a population divided by a physical barrier that prevents gene flow. Peripatric is when a small group breaks off from the main group and becomes isolated at the edge of the larger group's population range. So off to the side. Then you have parapatric, which is populations that are distributed across a continuous area but have limited contact in overlapping zones. So there's no complete geographic barrier. Maybe just individuals live far apart. And then sympatric is when new species arise from a single population without any geographic isolation. Whoop. Okay, I'm going to get caught up on stuff. So the analysis of genomes enables us to study evolution at a molecular level. DNA evidence may indicate how 2 species are related to one another, even if their body structures don't offer clues. It used to be all about structure. We would look at organisms, how they're structured, and go, they're related because they share similar structures. But now we can confirm it at a molecular level. So where do new genes come from? We know where new genes come from, right? You get some sort of a mutation. So one way in which new genes evolve is through duplication, then modification of existing genes. So synteny here, this shows you genes in common. All right. So these are genes in common with mouse and human. So you can see there is some degree of relation there, which makes sense because we're both animals, we're both chordates, we're both mammals, right? So there obviously is going to be some share of genetic information. We can also have copying of genes, where we have homologous chromosome exchange during meiosis in a process called crossing over. We learned about crossing over before. That could cause a new gene situation. And sometimes crossing over involves an unequal swapping of DNA, so that one chromosome in the pair of genes gets extra DNA. That extra DNA can carry part of a gene, a full gene, or a longer length of a chromosome. Then you can get duplicate genes. Sometimes copies of a gene undergo mutations that change their function. The original gene's still around, so the new genes can evolve without affecting the original gene or the product of it. And the first gene is duplicated, and then one of the 2 resulting genes undergoes mutation. So it's another way you can get a new gene into the population, new allele. Duplicate genes evolve as a result of mutations. So flow chart. And then gene families. Multiple copies of duplicated genes can turn into a group of related genes called gene families. Members of a gene family typically share similar yet slightly different proteins. So we've managed to group our genes together based on what they produce, right? What proteins they produce when they go through transcription, translation. So now let's talk a bit about molecular clocks. So how can we determine how closely something is related? So a molecular clock uses mutation rates, which they've been able to determine in DNA, to estimate the time that two species have been evolving independently. Something to remember. We did not like nobody is positing that humans evolved from chimps. That's not a thing. There is a branch where you can see chimps went this way, hominids went this way, right? So you can see, and we didn't evolve from Neanderthals, as some people like to think. In fact, we mated with them. That's fun, right? So we did not. So you have to think of this whole evolutionary process as more of a tree and less of a straight line, right? Because you have things that branch off and then further mutations occur over here. You have a branch that goes this way and there's further evolution over here, but it's not a straight line, but you can trace back to a common ancestor. So we have what we call neutral mutations that we use as ticks on a clock. So researchers use molecular clock to compare stretches of DNA to mark the passage of evolutionary time. This gets real techie. These are the guys that really like the math, okay, and the computer models. So the computer clock, the molecular clock, relies on mutations to mark time, and neutral mutations tend to accumulate in the DNA of species at about the same rate. So we know that mutations that don't cause any major impact on phenotype occur in a species at about the same rate. And we can take that rate and translate it into like a timeline and say, these are ticks of the clock, and we have had this many mutations since these two species split in their evolutionary line. It's kind of like radiocarbon dating, where you talk about half-life and you know how many half-lives an isotope has undergone within a certain number of years, so then you can trace back how old something is based on that information. It's similar to that. So comparison of DNA sequences between species can show how many mutations occurred independently in each group. So more differences that there are between the DNA sequences of two species, the more time has elapsed. So think of it like this. Think about the pendulum on a clock. It marks the time with the swinging pendulum, right? Tick, tick, tick. Oh, here's another Vecna reference for today. Tick, 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 tick. A molecular clock relies on a repeating process to mark time. Mutations, that would be the pendulum, your repeating process. Simple mutations occur all the time, causing slight changes in the sequence of DNA. Some mutations have major positive or negative effects on an organism's phenotype. These mutations are under powerful pressure from natural selection. Neutral mutations have no effect on phenotype and tend to accumulate in the DNA of different species at about the same rate. So we can now use this to estimate when there was divergence, when there was evolution occurring. So using molecular clocks isn't simple because there isn't just one molecular clock in a genome. Because some genes accumulate mutations faster than others, there are many different molecular clocks that tick at different rates, and the different clocks allow researchers to time different evolutionary events. Think of a conventional clock. If you want to time a brief event, you could use the second hand to time a long event. You could use the minute hand to time an hour, right? Researchers check the accuracy of molecular clocks by trying to estimate how often mutations occur, and then they compare the number of mutations in a particular gene in species whose age has been determined by other methods. That's how they check themselves, right? So now let's talk about body plans. This stuff is actually really kind of fun. Molecular clock's a little like, what? But this is kind of fun. So who remembers Hox genes? So Hox genes, yeah, these are your body planning genes, okay? Organisms have forelimbs and hind limbs, for example, and a spine, right? And a head. These are your genes that do that. So there's new research that they're calling this Evo Devo, which is great. And I love Evo Devo. It sounds to me like a 90s new wave English band, Anyway, it's because the studies in the relationship between evolution and embryologic development. So that's where you get Evo Devo. So Darwin himself thought that changes in the growth of embryos, like a mouse embryo, this one here in this picture, could transform adult body shape and size. And new research now shows small changes in Hox gene activity could produce the kinds of evolutionary changes we see in the fossil record. So how many Hox genes are involved in change? Small changes in a Hox gene activity during embryological development can produce large changes in adults. These aren't tiny, because these genes are so important in body segmentation and body planning that if there's a mutation in one of them, it's a hugely impactful mutation. So here's what? Jeez, gosh. So if you look here, here's your embryo basically developing into your adult species. So here's your larval. And you can see they know what parts of that are going to turn into what body segments of the adult organism, right? And we know the same thing about us too. Like we know how that functions with us. So Hox genes determine which part of an embryo develop arms, legs, wings. Groups of Hox genes also control the size and shape of those structures. Small changes in Hox gene activity during embryological development can produce large changes in adult animals. So here's your fruit fly at the top, and there's a brine shrimp at the bottom. And the difference, one of the big differences between them is that particular Hox gene, because it lost the ability between the brine shrimp and the fruit fly to produce additional limbs. It's got less limbs, the fruit fly does. Insects and crustaceans descend from a common ancestor that had lots of legs, lots of pairs of legs. Crustaceans such as brine shrimp still have lots of legs. Insects, however, only have 3 pairs of legs. And the recent studies have shown that insects, there was a mutation in a single Hox gene called UBX, and it turned off the growth of some of those pairs of legs. So you can see how that can cause a huge impact on an offspring. And because of mutations in a single Hox gene millions of years ago, modern insects have fewer legs than some modern crustaceans. Variant of the same Hox gene directs the development of legs of both animals. Each part of an embryo starts at a certain time, to grow at a certain time, grows for a specific time, and then stops growing at a specific time. So if you look right here, here's a human embryo, and there's your timeline for when different things begin developing, stop developing, or stop at that point. At least we're going to freeze for now. We're going to grow teeth forever, and we're not going to grow, upper limbs are going to stop at this point. They have been determined. So it shows you how that, now it doesn't mean they continue to grow. They do continue to grow. Obviously, your limbs grow after you're born. You get taller, you get longer. But the development of the initial structure is timed perfectly. And timing really matters. And we're going to do a really interesting experiment in just a minute that'll show you how timing really matters. So small changes in starting and stopping times can make big differences in organisms. So for example, small timing changes can make a big difference between long, slender fingers and short, stubby toes. All right, hold your hand upright, but flat. Look at it. Everybody do this. All right, look at your hand, look at your fingers. Don't stretch it or anything. Hold it nice and straight so you can see all of your fingers. All right, observe the length of your ring finger and your index finger. So this one's your ring finger, your pointer finger is your index finger. Okay, you see them? All right, now, if your index finger is longer, keep your hand up. Index finger, your pointer finger is longer than your ring finger. Keep your hand up.
Oh, these.
Yeah, compare these two to each other. Your index finger and your ring finger. If your index finger is longer, hold that up. Okay, now if your ring finger is longer, hold it up. Who has a longer ring finger? 1, 2, 3, 4.
It's different on each hand.
Is it different on each hand?
Yeah, for me it is.
Take an average.
Oh yeah, it's different from this.
Yeah, that's your
Wait, no.
It's like half and half.
Half and half? About half and half? Okay, let me ask you this.
Mine are like the same length on this.
On the same on that? Okay, if they're the same on one hand, but different on the other, if your index finger is longer, hand up. And then if your ring finger is longer, hand up. Okay, this is interesting. Usually it's a little more divided. So here's why. During embryologic development, the ratio of the second, which is your index finger, to your fourth finger is established by exposure to sex hormones like testosterone and estrogen. Typically, by the time you're done developing and on the opposite side of puberty, most of you are darn close, if not there, you will find that your higher testosterone, higher prenatal testosterone leads to a lower digit ratio, meaning the ring finger is proportionally longer than the index finger. Very often you're going to find men with a longer ring finger and women with a longer index finger. And that has to do with prenatal exposure to testosterone and prenatal exposure to estrogen. How much testosterone you have. This is one of those. There's a couple of things that you can do like that they're very minor little things, but it's interesting. So higher estrogen exposure results in a higher digit ratio where the index finger is typically proportionally longer. Mine is longer for sure. My hand is like very stereotypical, I suppose, even though I have stubby fingers. So still, yep. So the difference is called low digit ratio 2D40, and it's been linked to traits like athletic ability, endurance, certain cognitive and behavioral traits, and though genetics and other factors are also involved. So if you're somebody who had a longer ring finger, where are you? My longer ring finger crowd? It's different on each hand? So are you athletic? Do you have good endurance? I'm curious.
When I pull them back, the ring finger is longer.
When you pull them back, the ring finger is longer?
Or like
So yeah, so it's interesting. But very often you'll see like males, and especially your more athletic males, will have the visibly longer ring finger, which is pretty interesting. Pretty interesting. James has that. There you go. All right, so we have some extra time, so we're going to use it for review. All right, so I'm going to switch over. I got to switch over what screen is up here so the boys can see it. Now, what we're going to do is we are going to do sink or swim. So some of you have done sink or swim before. Everybody's waving at you, Yusuf and Eunice. All right, so the way sink or swim works is, okay, we're going to divide ourselves into two teams. So how about I have Dynamo and Lab team on in the front part here, and then how about I have Fibers and Ducks in the back part of the rim?
We should have just let me have some.
Why do we end up with this now?
This represents red leaves.
That's a better name.
Yeah, that's fine. So there will be sitting and standing. This is an active game. So if you guys in the back, you may want to come over to the kitchen so you can see better. It is because I'm awesome. So I have a Lego nativity. I bought pieces of it, but I made most of it because yes.
Friend who went flop.
I spent many years trying to find a nativity I could stand, and I didn't like any of them until I found the Lego one, and then I was like, "That's amazing." Ridley, do you want a chair? But yeah, pull a chair over. Okay, we got 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5. Perfect. 7 and 7. All right, Barrett, you're in the back, right, with them?
Yeah.
Okay, so rules for sink or swim. All right, here is how we play our favorite. I think this is our favorite review game, isn't it? Everybody who's played this before, we have lots of review games, but sink or swim always wins. So here's how you play. Divide into teams. Well, we did that. Stand in question answering order with your team. You'll be rotating. So you're going to be in a particular order. So Asher, you got to pick an end. All right, there we go. Question answering order. A student from the first team, which I will have you all be the first team, will be asked the first question. If the student gets the question right, that student will choose one of two options. Sink a member of the opposite team, which means you have to sit and remain sitting until you are unsunk. Sunk students, you can't answer or help answer. You're dead in the water. You're sunk. You can't answer. Or your other option is to unsink a member of your own team, which means that you tap them or say you're unsunk or whatever, and now that team member is active again. If the student gets a question wrong, that student is sunk, which means they get to sit in place and they don't get to answer any more questions. Now, your team can help you unless a member is sunk, then the member has to sit silently. Team is out when all members have been sunk. So when all the questions are answered or the allotted time has run out, the team with the most teammates standing is the winner. Only the person answering questions may speak. So nobody else, you can talk, but nobody else can give me an answer except the person answering. Teammates can give, not talk, but nonverbal cues, sorry. So you can give nonverbal cues. So it's almost like charades. So if you're sitting, you can't do anything. You just have to sit on your hands and bite your lip. But if you are, there's going to be, most of these are multiple choice or true or false. So you can give each other nonverbal cues, right? Like some people don't know sign language, so they'll do, 1 for A, 2 for B, 3 for C, 4 for D, that kind of a thing. Or they'll do, true or false, right? You'll have about 30 seconds to answer each question before I call you sunk. Are we ready? All right, we're going in answering order. Are you going first, Tyler? Okay, all right. You can go first. An organized plan for gathering, obtaining, and organizing information is what? B, scientific method, and he is right. So now you have no one sunk. So your option is to sink someone, and you get to pick who. Who are we sinking, Tyler? Asher, down you go. All right, now we're over here, Grace, which is not considered a branch of physical science. I can help her out.
Astronomy?
Wait, you can't talk. This is Grace's turn. Zoology, she says, and she's right. So now you can either sink or unsink. Unsink, Asher's up. Drew. Move this down a little. See, okay. Back this up a bit. All righty, Drew. Your independent variable is what you keep the same, true or false? False, he says, and he is right. So what would you like to do with your correct answer?
Think after, think after.
All right. Question number 4, Miss Claire. 1 kilogram is equal to 100 grams, true or false?
I'm pretty sure it's false.
She says false, and she is right. And what would you like to do?
Think Drew.
Think, Drew. Later, Andrew. All right, coming up over here. Now we are at Miss Kenna. Question number 5. An observation is made using senses. An inference is made based on prior knowledge and experience. True or false? True, she says, and she is correct. And what would you like to do with your correct answer? Blah, blah. She's down. And see, you were nice to her. Yeah. All right, so Colin, this would be you. The statement, the worm is 3 centimeters long, is an observation, he says, and he's correct. And you would like to? Bye, Barrett. Take a seat. Down he goes. All right. All right, who's up? Who is that? Caitlin? There you are, my friend. Okay. What's this? Oh, wait. Did I? No, that's it. Yeah. Desi, she says, and she is right. And you would like to? Unsync Drew. All right. So now who are we on? Lucas. What have we got, Lucas? Hecto, he says, and he is right. And you would like to? Unsync Asher.
You can take it.
All right. So who's up, Miss Ridley? All right. What's that one, Ridley? Millie, she says, and she is right. And you would like to?
No. We do it once. Then.
Game. It's so fun. All right. So next up is who are we on? Jordan? All right, Jordan.
D Senti.
Senti, he says, and he's right. And you would like to?
Right. Raise Asher.
Raise Asher. Okay. So who are we on? We got Xander? All right, Xander, what have we got? B, he says, Deca, and he's right. And you would like to? Goodbye, Asher. He is prepared. All right, so who are we on? Isabella? All right, what have we got, Isabella?
No.
A, she says, and she's right. And you'd like to? Aftersop. All right. So now we have gone back around. So now we're at Tyler. All right. A hypothesis is an idea that can be tested. Remember that a hypothesis cannot be proved or disproved. In order, I should say, for a hypothesis to be considered, it must be something unrepeatable or something unrepeatable. They're not helping. Say it again. Same. That's repeatable. What's the other thing it has to be? It's got to be one or the other. Oh, look, you're getting good clues. Different. Okay, we're going to go. It's going to be supported or rejected or supported. So good shot, but there you go. All right, so bouncing back over here. We have Grace. All right. Explain the difference. So she gets a big one between scientific theory and scientific law. We can help her if you can figure out how to help her. They're not very helpful, are they? They're trying, but they're like.
Nah, no, you're not getting the paper rolling.
Is proven
Close.
and scientific law is the same all the time.
Close. Real close. Scientific law tells what happens, a fact. Scientific theory explains why it happens, an explanation. So close but no cigar, but very close. All right, so she's down. So now we're up. Who are we up? Drew? Okay, 15, Drew. A common refrain these days, you get the big one. Is science is settled. Statement demonstrates fundamental misunderstanding of the nature of science. Why is it never settled? 3 points. I need them all. That's one.
Supported or a good report understanding one area is not mean a good report understanding the others.
That's two.
A long time.
Yeah.
Science is not about absolute truth, but knowing uncertainty.
You got all three, my friend. That's a good one. That's what happens when you've been around 8 years. This is his 8th year. 8 years of doing that. Very good. All right. So now we're coming to Colin. Question
He can
16.
make his decision.
Oh, sorry. Yes. What's your decision? You want to unsync Tyler, unsync unsync Barrett, or sync somebody here?
I think I'm gonna say solid.
Oh, that was smart because he's next. All right, Lucas, the process by which organisms keep their internal conditions relatively stable is called
Homeostasis.
homeostasis, he says, and he is correct, and you would like to?
Think after.
Oh. Being mean to his brother now. I see how this goes.
Think after.
All right. Kenna?
We need to make people.
An enzyme speeds up a reaction by lowering the active. Oh, sorry, by what? I was going to read all the answers. An enzyme speeds up the reaction by lowering the activation energy is correct. And you would like to? Oh!
Keep the thing.
All right, who is up? Jordan? What is the best answer? All life on Earth exists in?
The biosphere.
Right, and you would like to?
See, we need more people.
Yeah,
to
I'll
say
take
Really?
No. Sure thing.
Asher did not know today would be aerobics, but hey. All right, now who is up over here? Ridley? No, Caitlin. Caitlin. A group of different species that live together in a defined area is called a or an.
Community.
A community is right. And what would you like to do?
Yes.
They wouldn't do anything, but I was sunk immediately.
Yep. Oh, bye, Isabella. Down you go. She's like, what do I do? All right, question 20. Who's this? This is Lucas. Okay. The total amount of living tissue at each trophic level in an ecosystem can be shown in a what?
You saw.
Yep, she can't help. You can't help.
So
I'm gonna say B.
Biomass pyramid is correct. And you would like to?
Razor. You need Razor. We need Razor. We need people.
No.
I need lucky anybody. What if I don't? No, please. Yeah, I'm thinking about.
Unsync Isabella. Okay. So who are we on? Ridley? Is she my girl? Okay. The maximum number of organisms of a particular species that can be supported by an environment is called the
carrying capacity.
carrying capacity. And she is correct. And you would like to do what? Tyler is sunk. Barrett is sunk. Xander is sunk.
You'll have to go through Isabel.
Okay.
Question.
Number 22. Who is up? Drew, right? No, you're up. We're over here. You just went. You're up. All right. He gets to go. He gets to go. Are you ready, Asher? Are you sure? Are you sure, Asher? All right, Asher. Okay. Despite differences in size, shape, and function, at some point, all cells possess DNA and
A. I can't pronounce that.
organelles. Oh, you get sick again? No, he got it.
Yeah. Oh, Luca.
All right, so now we're back to Drew. All right, Drew. Question 22. What is used by cells to store energy required for cellular processes? cRNA. Down you go.
Yeah.
Down he goes. All right, so now we're on to Jordan. Is this you? All right, Jordan. different forms of a gene are called?
Wheels.
All right, very good. And you would like to? Raise Isabella. Okay.
Okay, look, I'm bad with names.
All right, Kenna, you're up. Kenna, which are haploid? Oh,
Is it multiple choice or one answer?
there's only one answer.
This says which. You need to think. Which are haploid?
A, she says, and she's correct. Would you like to raise somebody or sing somebody?
Okay. Take this.
I do.
Bye. Down you go.
Yeah.
All right, so now we're at Isabella, right? Isabella, you're up. Okay. The movement of water molecules across a selectively permeable membrane is called osmosis. True or false? True. And what would you like to do? Blair's up. Excellent. All right. Caitlin? All right, Caitlin. The first organisms to reproduce in a deforested area are known as a keystone species. True or false?
False.
False. What are they, by the way? Pioneer species, right? Okay, and you would like to what?
St. Clair.
Bye, Claire. Okay. So now who's up? Jordan again? All right. No pressure, you two. All right, Jordan. A natural resource that is easily and quickly replenished is known as a common resource. True or false?
Who said that?
You died.
False.
That's correct. What is it called? Renewable resource. Very good. And what would you like to do?
Raise pollen.
Raise Colin. All right. So who are we on? Ridley? Is this you, my friend? All right. Organisms with two identical alleles are homozygous. True or false? True. And you're right. And what would you like to do with that?
Please. Thank God.
There he goes. All right. All right. Jordan, no pressure, Jordan. Oh, is it? Sorry, Isabella? Okay, it's your turn. Taking advantage of naturally occurring variations in organisms to pass wanted traits to their offspring is called what? B, selective breeding, and you are right. What would you like to do?
Raise Claire.
All right, Claire is back up again. All right, so now we're back around. Is this Drew? No, this is Kenna. All right, Kenna? The process by which the code in DNA is copied into a strand of RNA is called? They can't help you. Just you two and Ridley. A, she says. Oh, no. Oh, no. Down she goes. Translation is when you're going from RNA to protein. Transcription is when you're going from DNA to RNA. Very similar. All right, Claire, is this you? All right, Claire. Which is an example of a vestigial structure in humans?
B.
B? She's giving me sign language. Yes, B, appendix. Very good. And what would you like to do?
Sinking. You need to sink. Yeah, sink someone. One person.
They have Ridley and Caitlin, and that's it.
You should sing.
Thank Ridley.
Oh, she's done already. So now, Caitlin, no pressure, Caitlin. All right, Caitlin, here you go. What's the best definition for scientific method?
You got this. You got this.
Read out the options.
Yes.
Too close together.
Yes. A, scientific method is a way of proposing an explanation for why a phenomenon occurs. B, scientific method is a process of forming an idea that can be tested. C, scientific method is an approach to seeking knowledge that involves forming and testing a hypothesis in a logical, systematic way. And D, scientific method is the process of forming and testing ideas. C is right. And what would you like to do?
Hey, someone. Sing someone. Sing.
She's the savior.
Come on, sing. Sing, man. Yeah, sing that right. What?
Drew's up. All right, so who's this? Jordan? We on Jordan now? The building blocks of proteins.
Amino acids.
Very good. And what would you like to do now? Oh.
Yeah, sing Drew.
Okay. All right. Again, Caitlin. No pressure, Caitlin. Which shows the correct order of organization for life?
Biggest, small.
Smallest to biggest. C, very good. You would like to?
I thought she said D.
I thought she said.
No, she said C.
She said.
She said C.
I won't.
Tyler's been down for a while. Barrett's been out the whole time.
Sing Jordan. Sing Jordan. You're good song. Yes, it. Yeah.
Bro.
Jordan wants out.
Come on, sing Jordan. Sing Jordan. I volunteer myself. Sacrifice. 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 Wait, let me
All right, what's your choice?
Raise hand. 2, 3, 4, 5.
Canada's up. All right.
Yes.
Now who are we on? Isabella, is it you? All right. Which best describes DNA in a typical prokaryotic cell? So it's giving you shape and location.
Aren't A and B the same?
the same thing.
Oh, yeah, they are. So that gives you a clue. Yeah? B, she says, and she's right. And you'd like to? Oh! All right. So, Kenna, no pressure. A or Ann is a trait that helps an organism survive and reproduce. What? We talked about it today. B, she says, and you are right. Oh. And what are you going to do with that answer? Drew's up. Okay. Question 40. Who are we? Claire?
Yeah.
All right. What did Lamarck believe?
He was the stupid guy.
It's the Lamarck guy.
Yo, she's consulting her nose.
No, she's not.
No.
Oh, Isabella is. That's fine. She's not answering a question right now. Oh, and this is for the win too, isn't it? Or the tie, because we're out of time. So
See?
see, she says she's right. So y'all win.
What?
Okay.
Remember, because he was the guy who was like, if you're buff, your baby's gonna be buff.
Yeah.
I thought she would stay.
Very good.
One bush.
We still had a whole bunch more that we could have done. Look. You want to answer one, Barrett? Here, for fun. Oh, no, we haven't gotten to that yet. Hold on. Or that. Hold on. Barrett wants a question. I have to find one we've actually covered.
So I didn't actually go out of.
There you go. Great job today. Study. You got it.
We don't talk about that. You heard nothing.
Outstanding.